2004, and then did a postdoc with Johan Akvist, after which he then started his own lab. So initially he started in Santiago de Compostela in Spain, and after a sabbatical at Scripps, he decided to move back to Uppsala, where he's now a senior researcher and docent. His main interest is in free energy calculations and GPCRs, and ideally the combination of both, which is also the topic of his talk. So Hugo, thank you very much.
Yeah. Thank you, Zoe, for the very nice introduction. Yeah, I slightly changed the title of my talk to reflect actually these two passions, which are the free energy calculations that we are discussing here, and the last applications that we have on GPCRs. So clicking here, okay. So moving to a brief introduction. The topic doesn't need introduction here, but still it's good to remind that with the gain on computational power, we are all used to see how we can virtual screen millions of compounds, but we are also slowly getting more used to have free energy perturbations and other free energy calculations in the pipeline of drug discovery. But also, this is a seminal paper from the late 80s from Peter Coleman, where the free energy calculations are applied in more fundamental biochemistry, like, for example, to examine the effect of mutagenesis data on the binding affinity of a substrate or of a transition state. So I will recover on this later, but also another topic that is discussed more technically is that, okay, when we do free energy perturbations, we probably don't think about continuum solvent representation. Some other approximate methods like MMPBSA, for example, do this, but normally when you see free energy perturbation calculations, you see like the use of infinite periodic boundary conditions. Now, I would like to remind that there is something in between, which are the finite periodic conditions, like spherical boundary conditions, to perform MD simulations that are suitable for free energy calculations. Now, in this setting, when you are thinking about a protein ligand binding problem, you embed the ligand in a bubble of water, in a drop of water, where you restrain this shell with a half harmonic attractive and Morse-like repulsive potential, and you perform totally unbiased and free MD sampling on the binding site. Of course, you have to select a big enough binding site for this. And you can imagine that this is the setup that I will use along this presentation. Now, what have we done in the last years at our lab for free energy calculations? Well, we have automated free energy perturbation pipelines, actually dividing this into groups. So one is the, let's call it classical ligand free energy perturbation calculations, which we call QILFEB, which you can think more for lead optimization, although I will show some other uses. And the other one is for performing in silico mutagenesis, or in general to evaluate the effect of a single point mutation on things like protein ligand binding, but also on protein stability. So this module is QILFEB. Both modules are freely available on the GitHub on this address here. Just change the pointer to, yeah. So let me start with the first one, QILFEB. This module was presented in the last chemistry conference by my then PhD student Willem Jespers, and I will briefly summarize the setup of this. So QILFEB is an API to automate free energy perturbation of ligands in our MD engine Q. So we want to automate, as Mark said yesterday, everything that gets boring for the user. And since free energy perturbation are, by definition, comparison between pairs of ligands, unless you go for absolute binding affinity calculations, which we are not there yet, but soon. So when you have two ligands, the first step is to generate the parameters, and we can do this we wrote here for 3 force fields. Also, the open force field is now implemented, so 4 force fields. And then, once you parameterize the ligands, you have to set up the protein ligand complexes. So you start with a protein ligand complex, and then you, since we said we run on spherical boundary conditions, you shrink the sphere, and then you do all the technical things. All this is made by the API, so the user doesn't have to take care of this. And then it comes the simulation, the MD simulation and free energy perturbation associated is done under the dual topology scheme. You run it in the bound and the reference state, and several replicas, which we normally do 10 per simulation. And then it comes the analysis module that does, well, a general analysis of the results, but also the statistical analysis. All this is described in this publication. Now, one thing that I forgot to mention here is that this is for a pair of ligands, but actually, when you have a series of ligands, you need to define the FEP pathway, the pathway to combine the pairwise comparisons in a way that you represent the whole data set. And this is an open question, not solved, and there are many ways of doing this. The chemical space of your data set can define this. Like, for example, in this case, the CDK2 inhibitors data set, you can see that it's relatively easy to compare all ligands with a reference ligand, because this is a series of congeneric series. Still, it can be problematic, and the results that we have shown here, you can see that there are 2 ligands that are a bit off. So the prediction is a bit, there's a bit of over prediction as compared to the experiment. Now, further analyzing this, we saw also that the 2 ligands that are a bit of an outlier also present a big standard error of the mean between the different replica simulations, meaning that the perturbation is not pretty much converged. So then an alternative way to show these ligands is, instead of comparing them to the central ligand, is to do a run going around and then do this pathway to connect the ligands. And in this case, the perturbations are much more converged, the standard error of the mean reduced dramatically, but also the mean unassigned error reduced in this case. So here you can see that this is the comparison that we made. So this opens the question, what is the optimal way to sample? In this case, we decided to do an all against all pairwise comparison, and from there to treat the data statistically to see what we can learn from it. So in this off-diagonal matrix that we have here represented the correlation between delta delta G experimental and calculated, we can see that some data are not well converged, but you can use this to apply some other algorithm like machine learning and collect the data points that are relevant. So we are now working on this, training this on different data sets, and actually we have preliminary data in this case with other comparisons like Morgan fingerprints here that will show the mean unassigned error of the data set from the one data set that was presented and discussed yesterday as compared to the reference FEP plus engine. And we see that we are doing slightly worse in some cases, slightly better in others, so we are getting there. So this will be presented more in the second version of this conference. Right now, I want to present the other part of the automated free energy perturbation methods, which is QILFEB. This is a single topology representation of annihilation of any side chain to alanine. So this way, one can perform an alanine scanning. But by combining 2 thermodynamic cycles, one can also compare a wild type with any other mutant connected by the central leg of alanine. Now you have to pre-generate the libraries for successive annihilation of all the atoms to alanine. I will not enter into details, it's described here, but the convergence with the recipes that we have implemented for each side chain is quite good, and we have extensively used for the site-directed mutagenesis analysis, first of the A2A adenosine receptor, where the comparison between the effect of a mutant to shift the affinity of agonists and antagonists is quite well correlated with experiment, but also to many other different, mainly GPCRs. Not only on G protein-coupled receptors we have worked. Here are the outline, the references. You can write me if you are interested in any of those. But also we can apply this to evaluate protein stability by fulfilling the thermodynamic cycle where the reference state is just a tripeptide unfolded around the residue that is mutated. And we can see that in this data set, T4 lysozyme, this is, the results are quite promising. Again, we will present this more in the second part of the conference. Now, combining QILFEB and QRES-FEB, combining the point of view of the ligand and the point of view of the protein, we can learn a lot. For example, we have shown such a combination for the A2A adenosine receptor recently in collaboration with Hysteresis, where we have assessed the growth and also predicted the binding mode of a ligand based on our free energy calculations in a way that correlated with the crystal structures that were produced at the end of this project. Now, I'm moving now to GPCRs. And I would like to explain some basics of the pharmacology of it, because from now on, we are not going to be focused only on predicting ligand affinity. We will aim to predict ligand efficacy. So what is this? Okay, so the receptor, the GPCR, is a machine to transduce the signal that comes in form of an agonist, and that way the receptor couples to the G protein normally and produces the dissociation of it and the transduction of the signal. Now, if this ligand is the agonist, it can be a full agonist that produces the maximum efficacy of the signal, or a partial agonist if the efficacy is reduced as compared to the one of the full agonist. So the partial agonist is not so efficacious as the full agonist. Now we have other types of molecules, which are the antagonists, that do the opposite. Either they block the receptor, and then it cannot be further activated via the agonist, or they stabilize the inactive conformation of the receptor, and they even reduce the basal activity. What is this basal activity? The basal activity is the capacity of the receptor to signal without any ligand. So the energy landscape of the receptor along the different intermediate conformations between the receptor without the ligand that stabilizes or the receptor with the ligand is different. And this is what can make us think, okay, we can correlate actually this with energies. And indeed, the first thing to look at is the pharmacological landscape of GPCR activation. This is from Terry Kenakin, but it's based on the theory from Bob Leibowitz on the allosteric effect of the G protein to stabilize the receptor, the active receptor, but also the agonist. So there are different equilibrium constants that connect this called extended ternary complex model. And if we translate this into structures, because we now have many structures of GPCRs, then we can think of this scheme where you have the inactive, the active-like receptor that has been activated or has changed the conformation towards the active receptor, and then that one with the G protein coupled. And this would be the agonist. Now, if we bind here an antagonist, then we can think of closing these legs on vertical, building the vertical legs and closing these cycles, because we can transform an agonist into an antagonist if they are chemically related. So this is the whole kind of way of thinking that we have to relate thermodynamic cycles with the pharmacology of GPCRs and predict ligand efficacy. So we are now trying to do computational pharmacology, and our test set will be, again, the A2A adenosine receptor, again, because it was already presented in this conference, and it is one of the receptors for which we have more structural information. We have different crystal structures in the inactive, active-like, and active conformation with different types of ligands. So the idea is to do free energy perturbation computational models to predict the conformational selectivity and to close this thermodynamic cycle that I just shown. So to get more into the chemistry of these ligands, the classical agonist of the adenosine receptors, the A2A and others, is a molecule that is similar to adenosine that contains a ribose that makes specific polar interactions in the binding site that promote the activation of the receptor, that promote the closing of this area and the conformation, the active conformation of the receptor. Now, the holy grail of agonist design on GPCR, on adenosine receptors, is to get rid of these ribose and to design molecules like capadenosone that was in clinical trials, or this cyanopyrimidine that have been demonstrated to be either full or partial agonists. Now, to understand how this works, we set up a FEP scheme where, well, since the receptor exists in a membrane, what we do is that to build the complex and equilibrate in a full membrane, but then we shrink a sphere and do local simulations on the binding site. And these are just the technical details. We can discuss on this later, the force field that we used, etc., and the lambda sampling, etc. Now, here, we don't simulate the transition of the inactive to the active receptor. We'll never do that, and we don't expect that because we do this in separate compartments. So we simulate the transformation of an agonist into an antagonist, both in the inactive and in the active-like conformation. And then we calculate the free energy differences, and by subtracting the vertical legs, we see what is the relative affinity of the agonist for the active conformation as compared to the antagonist, because the agonist should have more affinity to the active conformation than the antagonist. Then it is a more efficacious agonist. So this correlated well for this case, where you compare the agonist adenosine with similar antagonist without the ribose, and then we can kind of qualitatively see it, and that's the question, if we can qualitatively correlate the delta G calculated with the delta efficacy, in this case 90% loss of efficacy by removing these hydroxyl groups. Now we extended this to different families of agonists, full or partial agonists, and both adenosine and the related ligand NECA were compared to these antagonists without the hydroxyls, but also this series that we had designed a few years ago, that the ones that have this prolinol are partial agonists and the ones that don't have it are antagonists, or also this partial agonist from Leiden, where the position of this hydroxyl group shifts the efficacy of the ligand. So can we predict this? By doing the same for each pair of agonist-antagonist that I shown, then we correlated the loss in efficacy by the given chemical modification with the calculated free energy change of affinity of active, preferential affinity for the active conformation. And these are the three families of ligands grouped in boxes, as you saw before. So yes, we can do not only a qualitative, but a semi-quantitative correlation between the delta G, or the delta G, sorry, and the delta efficacy. Now, this is the binding mode of all these compounds, the ones with ribose, and where the other compounds put the analog that could do something similar to the ribose here. And we would like to see here what is the effect of some residues on shifting the efficacy of the ligands. Now, we are moving now to examine the effect of mutations on the efficacy of ligands, but this cannot be dissociated from the effect of the mutation itself on shifting the conformational equilibrium of the receptor. So if you have this equilibrium, there is a basal activity that the receptor spontaneously activates, and if you have certain mutation, it can have an effect, a detrimental effect on this. It would be a constitutively inactive mutation, or a positive effect, and it would be a constitutively active mutation, the one that stabilizes the active conformation. Now, what is this effect of this mutation on the efficacy of the different ligands that we have been discussing? Now we have some data for this, and we could join these two thermodynamic cycles. So first, for these two mutations that I mentioned before, serine to alanine in position 742, helix 7, position 42, or threonine in helix 3. So for the first one, we calculated by this thermodynamic cycle that compares the mutation in the active or the inactive state, that there is basically no effect. So the alanine mutant does not shift the equilibrium towards the active or the inactive. It's a neutral mutation. But this other mutation has a detrimental effect in activation and stabilizes the inactive conformation of the receptor. It's a constitutively inactive mutation. Now, if we put the other side of the thermodynamic cycle, we need to simulate a ligand binding to both the wild type and the mutant receptor. So what is the effect of the mutation of the efficacy on the ligand? We know that as well for these two mutations of, for example, this adenosine-derived ligand. So in this case, the threonine mutation has a big experimental fold change in the efficacy, and the serine has a lower change in efficacy. So by adding the corresponding leg and to the one that we already calculated, we see that we qualitatively and semi-quantitatively see this effect, that the threonine affects a lot the efficacy of this ligand CGS, while the serine affects, but not so much. The two, in this case, the two changes are on the same direction. Now, for this other partial agonist, the Leiden family of partial agonists, we see that the experimental fold change upon this mutation in efficacy, upon any of these mutations, is actually the other way around. Can we predict this, and why? Well, we see that calculating this leg here, it actually goes in the opposite direction. So the mutation increases the affinity of the ligand for the active conformation of the receptor. And this counterbalances the effect of the mutation, in this case, to be constitutively inactive mutation. For the mutation, we have more or less the same, and it also goes in the other direction. Since the mutation didn't shift so much the equilibrium towards the inactive conformation, then the overall effect is more like reducing, increasing the fold change, increasing the efficacy of the ligand. So as conclusions, I will say that the FEP protocols in Q are versatile, robust, and automated, and we can do FEP for ligands and for residues and combine it, and we can go beyond predicting ligand binding, and we can predict pharmacology of ligands, ligand efficacy for agonists and antagonists, and the effect of constitutively active and inactive mutations, and the effect or combined effect of this on the efficacy of the ligands even. Now, my last slide, as usual, is to give credits to the people that have done a bunch of work here, starting by Willem Jespers, who did his thesis on this topic, to Johan Åkvist, who is a professor at our department and is inspiring for all this and started all this research area, and David and Alejandro, who are working hard within the spin-off company that we have created, Team, in the case of Alejandro, and also the people that did the experiments, both in Leiden, the pharmacology experiments, Laura and Adam, all their people, and also Eddie Sotelo and his chemistry group in Santiago de Compostela. So I think that I'm ready for questions. Thank you.
Thank you very much, Hugo. That was a really exciting talk. Yes, we have a few questions here in the Q&A. So the first question is by David Ming. Have you investigated the hysteresis of alanine scanning?
Yes. Actually, here, actually, oops, it went off, maybe. Yeah, I want to show here, but the residues, when you mutated to alanine, it's much more stable than when you grow the residue. So we have a pre-computed hysteresis on the way back, but the real hysteresis we have tried, but growing a residue is much more problematic, because you never know where the side chain will be, and then you solvate. So in this case, it is something that usually does not converge, and we go for like annihilation, and that's why we have the protocol of annihilation. But the theoretical hysteresis of the successive annihilation, so at every step we can go backwards, and that is exactly what kind of pushed us to do this way of annihilation, where we first reduce the charge, then we transform the atom into a soft core atom, and then we do annihilation, and we do it for groups of atoms one after the other, and that is the way that we can achieve convergent hysteresis on each of these sites.
Thank you. So we have another question from Clara Christ. So could you comment on the pros and cons of the boundary conditions and the motivation to choose them? And as a follow-up, I have a similar question to that. I suspect this was done for speed up. So I would like to ask as a follow-up how you compare to other popular FEP programs such as AMBER or FEP+.
Yeah.
In terms of speed.
Yeah. So the motivation for doing spherical boundary conditions is, as you said, one is the speed, but the other one is also that you focus on the area of interest, and you are not having problems of long sampling. So yesterday at Francesca's presentation, she pointed out that sometimes you have some loops that move a lot, and then you need very long sampling times to kind of stabilize that movement and to neutralize the fluctuations that this can cause on your simulations or on your energies. So when you focus on the binding site, as long as you do a big enough sphere, we normally do like 50 diameter sphere as a default. Sometimes we do bigger. Then you avoid these kind of distant movements that are not affecting ligand binding, but are creating problems. So the other one, the speed, yes, it is true that you speed up the simulation, you reduce the number of atoms a lot. On the other side, our software normally does not scale the same number of processors that does AMBER or GROMACS. But that means that you can use all the other processors to do parallel simulations with different random seeds, etc. So the speed, since we have relatively short simulation times on top of having a smaller system, you need shorter simulation times to achieve convergence. And this is typically in the range of a few nanoseconds. And per ligand, the 10, of course, doing the 10 replicas, but since they are distributed, you can do 10, you can do 100, as long as you have processors, that's no problem. So it's normally between 2 and 4 hours, depends on the lambda sampling that you're doing per ligand.
Okay, great. Thank you. So another question is that GPCRs have known allosteric effects from different regions of the protein, including cholesterol binding and others. How can you model these effects if you confine the simulation to only a sphere? Is the rest of the protein rigid?
Yeah, so clearly on this case, you are not modeling these effects. So first of all, I didn't include these effects on my simulations. The way that you would have to do it would be to, again, build a thermodynamic cycle and connect the state with the allosteric or without the allosteric, with the conformational change done and without. You don't transition from one to another, but you compare the different states in stank compartments. So that is the way, if we can compare and if we can see the effect of activation without simulation the activation, of course, we can see the effect of allosteric modulation, but I didn't include this. But for example, I mean, in part I did, because the effect of a mutation is actually an allosteric effect. So when you mutate a receptor and you increase the basal activity or you decrease the basal activity, this residue is actually doing an allosteric effect. So,
but it is included in your sphere, of course.
Yeah, this was included in our sphere, of course. Yeah, but if you are having another residue, distal residue that does the same, but you have a simulation or a model with or without this, because it stabilizes one or the other conformation of the receptor, then you can compare one or the other conformation of the receptor. But yes, distal effects, correlation of the distal effects, then you have to increase your sphere.
Okay, and that's something that you can play with, right?
Yeah.
It's a parameter in the input. Okay. Kira
Absolutely.
wants to ask, Kira, thanks for the question. She wants to ask, which MD program are you using?
Yeah, so this is Q. It's only the letter Q. So it was originally written by Johan Oqvist like in the late 90s and his team there then, and now is maintained in an open source format by several groups here in Uppsala University. And it's available on, so my last slide is in a GitHub. So if you go for GitHub Q users, there's also a publication in X software where you can, it's not here, where you can see it. Yeah, it's freely available. It's open source.
Thank you. I'll ask one more question because we're running out of time, but it's very exciting. Hugo, have you looked into the trajectories of the active and inactive simulations to ensure that you're sampling the residues near the active site and the water network enough? GPCRs often have an important water network to conserve.
Actually, yes, the answer is yes, and I totally agree with this question. So in this case, I didn't discuss so much on it. We have, for example, on this other case where you have some mutations where a water molecule plays a role. Here it is shown one of these water molecules. Here's another one where some of the residues, the mutation of the residue disturbs this water network, and this actually has is where we see the effect in a case like this. We have plenty of examples where we have actually found the structural reason of the effect that we have seen on a free energy simulation to a water molecule stabilizing or destabilizing a water network. For example, in this in the one data set that it was discussed for different free energy perturbation methods, there is one of the targets that has one water molecule that is actually we are now actually, as I'm speaking, we are running the simulations where we are checking whether the disruption of a water molecule or actually an unstable water molecule that is displaced by one of the substituents is actually what is favoring the binding affinity. So yeah, definitely there is a lot of things on GPCRs and on any other target. I think that GPCRs are not so different to most of the targets. It's just that the structural biology was traditionally lacking, but now we have enough structures and some of them with high resolution. So yeah.
Excellent. Thank you very much for this talk. You still have some open questions.